So good morning, everyone. My name is Ilinca Sasu, and I worked on this project with my mentor, Lizzie Marr, and I'm currently presenting from Los Angeles. So before we jump in, I wanted to do this project because as someone with this condition, Crohn's disease, I understand the struggles that many patients go through, and I essentially wanted to help these patients by basically looking at this condition through a new lens, if you will. So let's get into it. So before we begin, I will just be covering, so since this was more of an experiment, I will be covering basically what Crohn's disease is and what CRISPR-Cas9 is, and then I will be going into hypothesis, then essentially how I did the experiment, and then, of course, the results, discussion, and conclusion. So let's jump into it. So Crohn's disease is an autoimmune condition that promotes inflammation in any part of the gut, and this can range anywhere from your rectum to the mouth. Now, Crohn's disease causes many symptoms such as severe abdominal pain, bloody stools, fatigue, and so many more symptoms that are essentially exacerbated depending on the severity of the condition. Now, given this condition, it's essentially very prevalent in the US. It affects as high as 2.8 million people, and this number is only rising. Now, biologics are a type of treatment used to treat Crohn's disease, and more specifically, TNF inhibitors. Now, TNF inhibitors are um, essentially types of biologics that neutralize Th1 cells that essentially promote inflammation in the gut that cause the symptoms by releasing these cytokines that essentially calm down the immune system. And the most popular and most successful TNF inhibitor used for Crohn's disease is infliximab. Now, as someone who's on infliximab, it has worked tremendously for me as it has helped me reach remission within 1 year of starting the treatment. However, as high as 30% of people are not so fortunate as I was. They don't respond properly to it, and thus their condition is essentially exacerbated. They have more symptoms and they face complications from it. And for these patients, typically surgery is their last resort. And surgery could be an issue because it's not essentially does not guarantee a better quality of life for the patient. Now, you may be asking, why was I lucky to experience remission with infliximab while others weren't? And this is where the ATG16L1 genetic mutation comes in. Now, the ATG16L1 gene is essentially this gene that is highly prevalent in lymphocytes and essentially differentiates between self and non-self in the immune system. However, when this gene is mutated, more specifically the RS224-1880 mutation, it essentially is unable to differentiate between self and non-self. So instead of attacking antigens and or harmful bacteria or viruses, it essentially just attacks everything, which actually promotes inflammation. Oh, here we go. So this is where CRISPR-Cas9 comes in. CRISPR is essentially a gene editing technique utilized by bacteria to edit the DNA of invading antigens. Now, the way it does this, it essentially cuts the mutated nucleotide in a target sequence and replaces it with, and either knocks out the mutated version of this target sequence, or it can replace it. There are multiple versions or ways that CRISPR can work on a target sequence. And it actually has been tested previously on leukemia and has been proven to work, which has given me a lot of hope that it could also be applied in Crohn's disease. Now, it has actually been tested on a different gene, the IL-10 gene, in a patient with Crohn's. However, it has shown little to no effect. Even though this was the result of the IL-10 gene, I still believe that it can be effective on the ATG16L1 gene, just given that it has worked previously in leukemia and sickle cell disease. So this is where my hypothesis comes in. I predict that CRISPR gene editing could help improve the effectiveness of medications for the treatment of Crohn's disease by reversing the ATG16L1 RS224-1880 T300A mutation associated with non-responsiveness to infliximab. So before we even dive into how I did this project, we first have to understand what this treatment plan or who this treatment plan would apply to. And first, we have to identify the different stages of remission or the progression of the disease, if you look at it as such. So first of all, clinical remission. This is essentially where a patient is showing no symptoms. They're essentially asymptomatic and their condition is dormant. Biochemical remission is essentially testing remission at a lab level, essentially looking into calprotectin levels and ensuring that they are below 120. And if a patient has calprotectin levels below this number, then they are essentially they would essentially be in biochemical remission. Third is endoscopic remission. Endoscopic remission is essentially testing for remission or progression of the disease of um, by essentially testing through procedures such as colonoscopies or endoscopies. Histological remission essentially detects inflammation at a molecular level. And lastly, surgical intervention or remission is essentially where a patient's condition has gotten so severe where it essentially surpassed the other four stages that surgery had to have been done in order to prevent the disease from causing more complications. So the population for this treatment to potentially apply to would be the 10 to 30% of people who are reactive, who essentially don't reach remission within 1 year from infliximab. And we would be testing first on people stuck in the first and second stages, so clinical and biochemical remission, before we move on to patients with more severe conditions. Then for the study, so to actually do this, first we wanted to focus on the ATG16L1 gene simply because of its prevalence in patients who are not responsive to infliximab and also who have the condition naturally. We also wanted to incorporate CRISPR software ChopChop, where it would then generate 157 target sequences. From there, I had to choose the best one based on three different factors that I will be discussing. And from there, then we were able to design a single guide RNA with ChopChop. So on to results. So based on the 157 target sequences, I first decided to look into 5 possible options. But before we discuss the 5, we first need to understand what the factors are. So first of all, efficiency. Efficiency is essentially the likelihood of and a single guide RNA reaching its respective target sequence. And the higher the percentage, the better. GC content is essentially the percentage of guanine cytosine pairings in a respective target sequence. And generally speaking, the lower the percentage, the better, as it allows for more stability. Then MM3s, which are essentially just the number of mismatches. Mismatches are essentially the number of incorrect locations that a single guide RNA could potentially go to. And typically is best to have minimal mismatches. However, the data that we've been able to collect has actually shown very surprising results. So as you can see here, rank 39 had the highest efficiency with as long as highest efficiency percentage as well as the lowest GC content. However, also out of these 5, it had the highest mismatches of 7. So even though it has the highest number of mismatches in the data, we decided to go with rank 39 as its efficiency ranking was significantly higher than many other target sequences in the data. And there were actually numerous other target sequences that have had efficiencies that have come very close to 74%. However, the number of mismatches exceeded 15, and we've even had target sequences with mismatches as high as 18, which is why we ultimately didn't go with those. It also had an ideal GC content. As mentioned previously, it's most ideal to have a GC content within the 40 to 55% range, and generally speaking, the lower the better. It also, as mentioned previously, it had a relatively low number of mismatches, as some target sequences in our data had mismatches as high as 200. So, of course, those were already going to be eliminated as a potential target sequence. So naturally in the data, we had actually noticed some outliers. For instance, rank 95, we refer to the target sequences as different ranks. So rank 95 had ideal conditions to be a target. It had a good GC content within the 40 to 55% range. However, its efficiency was only 4.51%, which was extremely low. So it was actually the lowest in the entire data set. We also noted that, so essentially this taught us that even though it has the ideal conditions to be an effective target sequence, it essentially was not. It just basically goes to show that even with ideal conditions, it does not necessarily guarantee a good outcome. We also noticed that a higher number of mismatches did not necessarily correlate to lower efficiency rates, as rank 39 being an example of this. We also noticed that rank 112 had an efficiency of 75.06%. However, it had 18 mismatches. So that was ultimately why I did not go with that, because the number of mismatches at that point was just simply too much of a risk. And I will be discussing why mismatches are such an important factor when considering target sequences. But before we get into that, we also have alternatives. Now, as mentioned previously, we also need to consider alternatives to different target sequences in the event that if this project were to be applied in the lab and rank 39 just does not work, we could also potentially test on rank 47. It has it has fewer mismatches. It also has a really high efficiency, as high as 68%. So that also gives us some perspective on a possible alternative to rank 39 in the event that it doesn't work. So, as mentioned previously, mismatches are a major issue to be considering when applying this treatment plan in a lab. So, first of all, we need to consider off target effects. Now, off target effects are essentially what happens as a result of the single guide RNA reaching the wrong location and cutting out the wrong part of the sequence. And when this happens, it causes more mutations. It could cause an insertion or deletion in the sequence, and this can cause numerous mutations and more complications. And after all, we do want the patient to get better, not worse. So this is a major thing to consider. We also note that efficiency is very important to consider. However, it also goes very hand in hand with mismatches, as noted by the fact that higher efficiency doesn't necessarily indicate lower mismatches. In fact, rank number one had no mismatches, so it would ideally be the best target sequence, and yet it had an efficiency of only 41%, which is extremely low. Now, ultimately, with this, we know that there is a good chance that CRISPR can be effective, just given all the factors and given the implications and essentially risks of this, there is a good chance that it can be effective. So to summarize, we hope to test this treatment plan on people who haven't reached clinical or biochemical remission first, and essentially the people who are reactive to infliximab. And the end goal of this is to make patients who previously were reactive to infliximab become responsive to it and help them reach remission. However, there are some limitations. First of all, we need to consider funding and then necessary resources for a project like this to be carried out. And we also, again, need to consider side effects from off targets, as these actually increase your risk of cancer and your progressing Crohn's disease. Thank you for listening. Any questions? And these are again my references.
Amazing. Thank you so much.
Thank you.
Just even, well, so I just have to say, I feel like CRISPR is something I hear a lot about.
Mm-hmm.
It's kind of it's well known, it's mentioned a lot of the mentors that I work with, they use it. It was so cool for me to like actually see it in action though, and like especially that one slide and the visual you had. That was so cool to really like yeah, understand this concept that I kind of like hear and vaguely am a understand. So thank you so much for kind of giving me that gift and to just kind of really breaking it down like that.
Yeah,
Um,
thank you.
just even, I feel like your last slide kind of referenced this too. But I'm curious kind of what is your hope for continuing to work on this? Obviously your interest in your relationship with Crohn's disease is ongoing. What are kind of like the next steps that you hope to build upon this and keep pressing forward kind of even more of like an advocacy or better understanding of this treatment option.
I would love to test this in a lab and just see if it actually works first. But before we even test on
Yeah.
humans or even animals first, we would have to first test on like on a cellular level and
Mm.
hope and ensure that it actually does work. It does show improvement of the disease before we can even move on. So I would love to try to just apply this in a lab and just see how it works and then just tweak it from there. Because I do strongly believe that this method could actually work, given that it has worked previously in sickle cell disease and leukemia and has actually historically been tested on another gene in Crohn's. And even though that had different results, I believe that with just the right gene targets and the right application, it definitely could work, which is why I want to really like just expand on that.
Yeah, okay, amazing. Thank you so much.
Thank you.